The first step of eukaryotic translation is initiation.
First, the small ribosomal subunit will bind to the initiator tRNA carrying an amino acid.
That complex then attached to the 5' methylguanosine cap and began to scan for the AUG start codon. Initiation factors that were present include the EIF4 and the poly A tail binding protein that help the EIF4 bind to the tail of mRNA that was on the 5' cap.
The large ribosomal subunit attaches to the complex, allowing for the EIF1, EIF2, and EIF3 factors to be released.
The large ribosomal subunit contains 3 sites. The A site is the entry point.
The P site carries the peptidyltransferase that will eventually carry the polypeptide chain. The E site releases the now dissociated tRNA in order to make room for the next tRNA down the line.
GTP allows energy for the amino acids to bind the tRNA to the A site of the ribosome. The tRNA must then go through codon-anticodon pairing.
While this is happening, the tRNA in the P site releases the amino acid it's carrying. tRNA goes from the A site to P site to E site and continues these steps with new tRNAs to make the polypeptide chain as the ribosome moves down the mRNA strand.
The last step of translation, called termination, begins with the recognition of a stop codon. A release factor will bind to the mRNA at the stop codon. This will cause the polypeptide chain to be released and the ribosome moved from the strand.
Bacterial translation is similar to eukaryotic translation. To begin, bacteria need initiation factors IF1, IF2, and IF3. IF3 binds to the small ribosomal subunit, which then binds to the mRNA strand, upstream of the start codon at the Shine-Dalgarno sequence.
IF1 binds to the small ribosomal subunit at the A site. IF2 escorts the initiator tRNA where it binds to the P site. Codon and anticodon pairs are needed in order to bind.
Once the initiator tRNA is placed, IF3 is released, and the large ribosomal subunit takes its place on top of the smaller ribosomal subunit. Once in place, the GTP releases the rest of the initiation factors.
EFTU, an elongation factor, binds to the aminoacyl tRNA and enters the A site. The amino acids then bond to one another and the EFTU is released.
Next, EFG binds to the ribosome. The GTP bound to it gives it the energy to move the 2 subunits down the mRNA strand, opening up the A site. The uncharged tRNA is now located in the E site and expelled. These steps repeat until a stop codon is reached.
A release factor, RF1 or RF2, cuts the bond holding the peptide chain to the ribosome, which releases it. RF3 comes in and causes the other release factor to be expelled.
Finally, a ribosome recycling factor comes on to undock the ribosome from the strand. IF3 reattaches to the small subunit to keep them from reconnecting. Coronavirus has spiked proteins that infect eukaryotic cells, attaching to the mRNA. Then the viral proteins are translated along with the eukaryotic mRNA and become expressed, which is what causes people to become sick.
When coronavirus proteins bind to bacterial mRNA, endotoxins are released by the bacterial cells, causing COVID-19 symptoms like fever and inflammation. The COVID-19 vaccine is an mRNA-based vaccine. Simply put, the vaccine mRNA is translated and teaches our cells to make a protein to create antibodies against the actual virus.